be talking about this morning for me, for afternoon for you, is how we can generate reliable scientific and specifically medical evidence from what I will refer to as real-world data. I'm a professor of biostatistics, and I realized that I use a lot of biostatistics and epidemiology and medical jargon. And so one piece of medical jargon that I wanted to make sure that I defined before I dive into my talk is real-world data. So recently, this concept of real-world data, or data that are generated essentially as a secondary product of the fact that many of our transactions are recorded in a digital way, has become sort of a high priority for conducting research, especially medical research. And what I'm going to start off with are a few titles of editorials and perspective pieces from medical journals that came out in the pandemic in response to the rise in the use of real-world data to try to answer questions, medical research questions, in a timely way using so-called real-world data. So here's the first one. It says the pandemic is prompting widespread use and misuse of real-world data. So what we saw early in the pandemic was a situation where medical providers, clinicians, were faced with a need to make treatment decisions in an almost total absence of evidence about the relative effectiveness of different therapeutic options for treating patients with COVID-19. So what people started to do was use electronic health records and medical claims data to answer those questions, because the data were readily available. We didn't have to wait for clinical trials to be conducted. We could just move ahead and start to look at outcomes of patients receiving one treatment versus another. And so there was a real uptick in this type of publication. And then almost immediately we started to see responses that looked sort of like this, pointing out that there are real challenges and pitfalls to using real-world data to generate medical evidence, and that we have to be pretty careful about how we do it in order to avoid misuse of these data. Here's a second one. Can real-world data really replace randomized clinical trials? So there's always been this sort of tension between observational studies and clinical trials, with observational studies seen as providing a lower quality of evidence. But in particular, observational studies using real-world data raise additional questions. And some people started to say, well, maybe we shouldn't be doing this. Maybe we should, go back to just focusing on clinical trials, and using real-world data is really leading us down the wrong path. And then I have this one, which was a editorial in the New England Journal of Medicine, The Magic of Randomization versus the Myth of Real-World Evidence. So this was a very high-profile piece that generated a lot of consternation among people like myself who do research using electronic health records. And in particular, I think this is a somewhat problematic perspective, because it promotes this dichotomous thinking that somehow randomized clinical trials are magic and real-world evidence is just a myth. And in fact, you can do a very poor clinical trial, randomized clinical trial that provides rather weak evidence. You can also, in my opinion, do a very good study with real-world data. So what I'm going to talk with you about today is some considerations at each stage in the design and analysis of medical studies using primarily electronic health records data, in order to try to ensure that we can use these data to address questions that would be hard to answer using primary data collection or using clinical trials. Do so in a timely fashion so that we can start to respond to this urgency when we do have questions where there really is a need for timely generation of evidence, and avoid some of the pitfalls that would lead us to misuse of real-world data or the myth of real-world evidence. So the final title that I'll leave you with is this one: Randomized Controlled Trials versus Real-World Evidence, Neither Magic Nor Myth. And that's really the perspective that I'm going to take today. I'm going to talk about how we can do our best in terms of principled design and analysis of medical research studies in order to try to capitalize on the promise of real-world data, certainly recognizing that there's no magic, but I think there is a real potential for generating useful and valid medical evidence. Specifically, in my talk, I'm going to focus on 3 key areas that I think we should pay attention to when designing and analyzing medical research studies using EHR data in order to try to ensure the validity as well as timeliness of the evidence that we're generating. So the first part of my talk will focus on the concept of fitness for use, and making sure that our research questions are appropriate for these data sources. And I'm going to give an example of how data provenance, EHR data provenance, can guide the choice of measures in our studies and how we identify data elements from EHR. And then finally, I'm going to talk about information bias and how our methods should be tailored to acknowledge the presence of measurement error and misclassification in EHR-based studies. All right, so the first thing that I'm going to talk about is this concept of fitness for use, focusing on the question, is EHR data fit for my purpose? Fit for use or fitness for use is a very common concept that just means the data that we're analyzing should be of appropriate quality for the use that we intend to put them to. So I frequently talk about doing research with electronic health records to a variety of different audiences. And I found that with statisticians in particular, people are often very concerned about the quality of the data. And I've encountered a number of statisticians who respond to the idea of doing research with electronic health records with some dismay or horror, and have said things to me like, I wouldn't touch EHR data with a 10-foot pole. I think that lumping all EHR data or all real-world data into this category of messy data or bad data is inappropriate, because all data have imperfections in one respect or another. No data set is perfect, with no measurement error, no misclassification, no missing data, no confounding. All data sources have some challenges. And the question is really whether the data source that we're planning to use is appropriate for our purpose. So when starting to think about advancing the medical research base using EHR data, I think the question that we should be focusing on is not whether or not EHR data are good or bad, or whether real world evidence is magic or myth. The question is really, are EHR data appropriate for the use that we are intending them for? Additionally, as I move into later parts of my talk and start to think about different methodologic issues that we need to address in the analysis of EHR data, the importance of those methodologic issues is going to vary depending on the specific question that we're trying to answer. And there are some questions that we can answer with very straightforward methods in EHR, and there are others that are going to require a lot of really careful methodologic thinking and possibly new methods development. This whole concept of real-world data and real-world evidence became popularized in the US a few years ago after Congress passed a bill called the 21st Century Cures Act. So the 21st Century Cures Act said that the FDA had to develop a pipeline for incorporating real-world data into the product development pipeline, or the product approval pipeline. So because they were mandated to do this, the FDA started to think really carefully about what a rigorous regulatory quality level analysis of real world data would look like. And of course, people got really, really excited, particularly pharmaceutical companies got really excited about this possibility, because if they could potentially get a new product approved using EHR data, that would save them a whole lot of money versus conducting a clinical trial. So one of the things that the FDA did in their published framework for real-world evidence was to describe the many different ways in which real-world data could potentially be part of the medical product development process. So here I have listed a bunch of bullet points that are the different uses of real-world data, primarily EHR data, that the FDA identified. And these are to some extent ordered from the lowest risk to the highest risk of bias types of activities that we might engage in. So, if we're doing hypothesis generation, if we're doing exploratory risk factor explorations, identifying prior distributions for Bayesian models, those tend to be lower risk of bias activities, primarily because they're going to be followed by subsequent activities that are going to use methods with lower risk of bias are potentially more rigorous. When we get down to the bottom of the list, we start to think about generating evidence on product safety and particularly product effectiveness. This is really a challenging thing to do with EHR data, and it has been done to a limited extent so far in some special cases like oncology drug development for very rare cancers with no treatment options. But for the most part, the FDA has been really, really cautious about allowing people to use EHR data in demonstrating the relative effectiveness of different medical products, I think for pretty obvious reasons. Nonetheless, this question of the relative effectiveness of treatments and trying to figure that out using EHR data is something that people really would like to be able to do. So as I said earlier, early in the pandemic, clinicians didn't know how to treat patients coming into the hospital with COVID-19. There was a real absence of evidence and an urgent need. And so using EHR data, there are many studies that were published looking at the comparative effectiveness of different treatments and trying to use best practices for statistical analysis, for confounder control in particular, to generate valid evidence. So this is an example of one such paper which looked at the comparative effectiveness of hydroxychloroquine and azithromycin for treatment of COVID-19. This was using data from a healthcare system in the US, the Henry Ford healthcare system. I think they used reasonable methods to try to control confounding by observed confounder variables. They used a propensity score approach to address that. And in this study, they came to the conclusion that hydroxychloroquine was effective for treating COVID-19 and performed better in terms of reducing severity of outcomes than azithromycin. So subsequently, many additional studies have been conducted of the same question. I think the clinical consensus is that hydroxychloroquine is not effective and really doesn't do anything for treatment of COVID-19. So we could think about this as a study that essentially came to the wrong conclusion. And I would argue that the reason that this study came to the wrong conclusion is because this is an incredibly difficult question to answer using EHR data. So looking at the effectiveness of a treatment for the indication that that treatment was prescribed for has a very high risk of bias due to confounding. And even if we use state-of-the-art, best-in-class methods for confounder control in a setting where there is a high risk of confounding and EHR data contain very sparse and incomplete information on confounder variables, it's just really, really tough to do a good job. So that kind of question is not necessarily a good fit for analysis with EHR data. In comparison, this is an example of a less exciting but better fit for purpose study that a group that I was collaborating with at University of Pennsylvania conducted using EHR data, also related to the question of hydroxychloroquine for COVID-19. But we asked the much simpler question, which was just how did prescribing patterns for COVID-19 change before versus after the US federal government, in the form of former President Trump, endorsed its use for that treatment. And what we observed, not surprisingly, is that there was a huge spike in prescribing of COVID-19 after that endorsement. But this type of medical research question, which is referred to as utilization study, is much lower risk of bias using EHR data, because we know that the pieces of information that we need in order to answer this validly are reliably collected in EHR. A utilization study only requires that we have good information on what was prescribed, depending on specifically what we're going to ask, perhaps by whom and to whom and when. So this is just a fundamentally easier question to answer using EHR. So that's really all that I have to say about fit for use, just that it's important for us to think about, and importantly, when we're evaluating whether or not a question is going to be fit for use or whether EHR data will be fit for use for a particular question, the thing that we need to be reflecting on is which data elements are captured in the EHR, and are they adequately and systematically captured. So in the next part of my talk, I'm going to get a bit into data provenance and issues of the heterogeneity of the data elements that are collected in EHR data. But in general, if a piece of information may discretionarily be recorded in the EHR, but it's not required for treatment or billing, it's not something that we should really be basing our analysis on. It's generally easier to answer utilization questions using EHR than comparative effectiveness questions. They're generally going to have lower risk of bias and hence generate more valid evidence. Of course, we also have to actually be interested in answering those questions in order for that to be a good idea. And if we are interested in comparative effectiveness questions, asking questions that are orthogonal to the visit process or the disease process can help to limit challenges that arise, such as confounding by indication and informative observation issues. And what I mean by that is when we ask a question about the outcomes of a treatment for the indication that that treatment was prescribed for, there's a very high risk of confounding by indication. But if we ask a question about the outcomes of a treatment that was prescribed for something else, there's a much lower risk of confounding because the confounders that would be relevant were not in the mind of the clinician when they made the treatment decision. So for instance, I look at studies of drug repurposing, where we're interested in whether, say, drugs for treatment of diabetes have an effect on cancer. Well, since the patient's cancer severity was probably not in the mind of the clinician when they made a prescribing decision regarding which diabetes treatment they should receive, the confounder issues are much less acute. But finally, some questions are just really, really tough to answer with EHR. And I think it's important that we acknowledge that there are some things that we just are not going to be able to answer and be willing to say, this is not fit for use with EHR. This is a setting where real world data are not going to generate high quality real world evidence. And we need to think about doing primary data collection. So keeping in mind that we first have identified a study question that fits with the EHR data, the next thing I'm going to talk about is how we can derive measures from EHR data that appropriately acknowledge its provenance. On this slide, I have a figure that comes from Google Labs, where they are illustrating the process of a single patient interacting with the healthcare system. So here we have one patient who over time has a series of different kinds of visits, a primary care visit, here later in time they have a hospitalization. And at each of those points in time, they have different pieces of information recorded. So sometimes they have laboratory tests performed, they have medications prescribed, at various points in time they have diagnoses that are recorded. If we think about taking this, this process of a patient interacting with the healthcare system and transforming it into a database, it's going to give us a data set that looks essentially like data from any longitudinal observational study. So we have repeat measures for a subject of various different kinds. However, unlike any longitudinal observational study that we would have designed, there was no prior specification of timing of the visits, which data elements were collected at each visit, or even how those data elements were defined. So we might have one provider who assigns a type 2 diabetes diagnosis using one set of criteria, and another provider who assigns the same diagnosis code using a different set of criteria. So we could take these data that we've formatted into our longitudinal observational data set, we could stack them together with all the patients from our healthcare system, and we'll have a big observational database that we could then use to study all kinds of questions. But it's going to be very, very heterogeneous because across patients, there's going to be a lot of variability in how frequently the patients are seen and what's recorded when they are seen. So this kind of complexity introduces a lot of challenges from a statistical perspective. For one thing, because we have a visit process here that violates the assumptions of many standard statistical approaches. We typically assume that the observation process and the disease process are independent, and in EHR data, that's going to be very much not the case because most of the time people go to the doctor because of a health complaint. This is a schematic illustrating what I think of as the EHR data provenance process, which is essentially a filtering down of the information that we ultimately end up with in the electronic health record. I think that people who don't have hands-on research experience using EHR data have a little bit of a misconception that EHR data represent the health of patients. And in fact, EHR data are a very imperfect representation of a subset of the population that we really want to be studying. Because firstly, in order for data to end up in the EHR for us to study, the patient has to have received care within whatever system or set of systems are generating data for us. The patient has to have had an interaction with a clinician that results in documentation of the information that we're interested in. So one of the big challenges with EHR data is that often information that we're interested in might be recorded in a narrative text note. For instance, if we really care about things like behavioral risk factors like smoking and drinking or exercise, it's definitely possible that a clinician could write those things down in a narrative text note, but they don't have to. If it's not required for administration of clinical care or billing, then it's going to be discretionary whether or not it ends up there. And we introduce bias if patients who have that information recorded are systematically different from those who don't have the information recorded. So we lose information that's just not documented or that wasn't part of an interaction that took place between the patient and the healthcare provider. These are also huge, messy databases. And so there is frank error that occurs of various kinds. In any sufficiently large database, every error that can occur will occur at least once. And there are also just undesirable features such as temporal variation in coding practices for certain conditions in response to things like reimbursement policies. And we don't want those things to really influence the information that we have about patients' health, because it doesn't reflect changes in patients' health. It reflects differences in reimbursement policies, but it ends up in the EHR anyway, and it's really difficult to tell the difference. So I'm going to give you an example of a methodological project that I worked on developing an approach for EHR phenotyping. So EHR phenotyping is a really broad term that refers to the process of extracting data elements from the EHR. Most of the things that we care about and that we need in order to conduct our research, most of the pieces of information are not recorded in a gold standard form in EHR data. So quite often, when we're conducting a study, either for our outcomes or our exposures or our confounders or all of the above, we need to go through some kind of phenotyping exercise to decide how we're going to define the characteristics that we're interested in using the data that we have at hand. However, because of the heterogeneity of the interactions between patients and the healthcare system and the complexities of that data provenance process, we're going to have really irregular data that's different across each patient available. So it takes a little bit of finesse to get valid measures out of the EHR. Most commonly, this process of EHR-based phenotyping relies on what's called clinical decision rules. Clinical decision rules is a really, really simple approach to phenotyping that basically says we're just going to use expert knowledge of the condition and how it's coded and come up with a deterministic algorithm that classifies patients according to whether or not they have a particular set of codes or other data elements recorded. So these can be really, really simple. The very simplest one, which anyone with experience in medical research, I'm sure, has seen, is we looked for at least one diagnosis code for the condition of interest. And if the patient had that diagnosis code, we classified them as having the condition, otherwise not. Diagnosis codes in EHR tend to be, at least for some conditions, not particularly sensitive and also not particularly specific. So often we'll use more complex algorithms that require presence of multiple pieces of information, maybe a diagnosis code as well as a code for a treatment, usually used to treat that condition. They may also include time components, but in general, this is a pretty straightforward way to do phenotyping. However, it has some limitations. So this is an example of a rule-based phenotype for type 2 diabetes that was developed by a group called the eMERGE Consortium, who brought together data from several healthcare systems across the US, which were used for phenotyping, along with genetic data for those patients. So they were able to look at genotype-phenotype relationships. In this algorithm, the way that it works is everybody enters up here at this top node, And as long as the answer to each question satisfies the condition on the path to the next node, the patient continues to move through the algorithm and is potentially classified down here at the bottom as type 2 diabetes case. As soon as you don't satisfy the condition, you're kicked out of the algorithm and assumed to be not a type 2 diabetes case. So this is a little bit complex, but it's very common sense. We're basically going to look at the patient data that we have, see if patients have diagnosis codes for the condition that we're interested in, see if they don't have diagnosis codes for other conditions that might be confused for the condition we're interested in. We look for treatments, we look for abnormal biomarker values. It's a very common sense kind of thing to do. So the challenge with applying this kind of algorithm in EHR data, given the heterogeneity in data available across patients, is that this doesn't take into account the depth of information that's available across patients. So if we have a patient in our healthcare system who had one encounter one time, it had absolutely nothing to do with diabetes, and so they don't have any of these codes or medications recorded, we would classify that patient as not having type 2 diabetes. We might have another patient who has a whole history of many encounters with our healthcare system, including visits where their glucose or hemoglobin A1C was measured and found to be in the normal range. And that patient also would be classified as not having type 2 diabetes. But our diagnostic certainty for those classifications are extremely different. And this doesn't reflect that difference. We're not able to capture differences across patients in the certainty or the quality of our classification. An additional challenge that arises in EHR phenotyping is the risk of missing not at random missingness mechanisms in availability of certain data elements. So the example that I'm using here is availability of biomarkers for making that type 2 diabetes classification. So if we had access to biomarkers, including fasting plasma glucose, random plasma glucose, or hemoglobin A1C, that would be the best way to make a classification as to whether or not a patient has the condition of interest. But only some patients will have that information recorded. And what determines whether or not that information gets recorded is related to all sorts of characteristics of the patient. So in some cases where receipt of, say, a diagnostic test, like a biomarker, is just associated with whether or not the patient is in a high risk group within our population, that might represent a missing at random missingness mechanism. So for instance, if risk factors like age and race or clinical characteristics that are routinely recorded, like BMI, are associated with availability of the biomarker, and these things are observed and measured and well captured in EHR, then we would have a missing at random missingness mechanism. However, typically data provenance is actually much more complicated, and healthcare seeking behavior, which I would consider to include all sorts of things like health literacy, healthcare access, health insurance status, and so on. Those things will also be associated with whether or not the patient turned up, got evaluated, was willing to go have their blood drawn, and so on. And really importantly, as I said earlier, the EHR is a very imperfect reflection of what is actually going on with patients' health. So the characteristic that we're trying to determine here is type 2 diabetes. But patients with that condition of interest are almost certainly more likely to have their biomarkers assessed. And even if there isn't a direct connection between this underlying latent condition and measurement of the biomarker, there are almost certainly all sorts of symptoms of this condition that are known to the patient, known to the clinician, influence whether or not the patient has their biomarkers assessed, and yet are poorly recorded in EHR, if at all, further leading to a relationship between this unobserved characteristic and measurement of the biomarker, and hence a missing not at random missingness mechanism. So the approach that my group developed to try to address some of these challenges with phenotyping that I've just summarized was a latent Bayesian phenotyping model. And I'm going to summarize the features of this approach on this slide. So the idea behind this approach is firstly to acknowledge that the true phenotype is unobserved for everyone. Everyone has a true health condition, that we don't have direct access to in the EHR. And here I'm going to denote that Y and assume that it follows a Bernoulli distribution where each patient has their own patient-specific probability of having the phenotype theta I. So theta is the thing that we really care about. That's the individual patient's probability of having the condition of interest. We don't observe the true phenotype itself, but we do observe a whole bunch of features related to that phenotype. So these would be things that are routinely collected in EHR data, like diagnosis codes, receipt of prescription medications, various biomarkers, and so on. And each of those features we assume follows some distribution that I'm just generically denoting D. And each distribution has a set of parameters mu k, and they're indexed by k to represent the fact that the parameters of that distribution will differ depending on which of the latent phenotype classes the patient is in. So for instance, if we're talking about something like a diagnosis code, it makes sense that you're more likely, so you have a higher probability of having that diagnosis code if you have the phenotype compared to if you don't have the phenotype. We additionally allow missingness in the features to be a feature itself. So for instance, with diabetes, we think that patients who are getting their glucose measured are more likely to have diabetes. So presence or absence of a biomarker would also represent a feature that we could potentially use to try to deduce who has this underlying phenotype of interest. We did estimation in this case using a Bayesian approach, which was particularly nice here because using a Bayesian estimation framework allows us to incorporate information from the scientific knowledge base into our model. So specifically, some of the parameters in these distributions for our features, we have pretty good prior information about because we know things like the diagnostic accuracy of glucose or hemoglobin A1C for type 2 diabetes. So we can encode that information in the form of a prior distribution that says, hey, here's how big the shift in that biomarker is expected to be for a patient with versus without the phenotype of interest. So we're going to take the priors that represent what we know about these features based on the medical knowledge base. Our likelihood takes the form of a mixture likelihood where we're mixing over the two unobserved phenotype classes. And that's going to allow us to estimate the posterior distribution for theta I. So the posterior for the probability that each patient has the phenotype of interest. The nice thing about this is rather than making a binary classification, which doesn't really reflect the fact that we have a different amount of information for each patient, we're now going to get a probability. So for some patients, we may be able to assign them a probability that's quite close to 0 or quite close to 1 because we have very strong information telling us one way or another whether they have the phenotype. And for other patients, they're going to be somewhere in the middle or somewhere closer to the population prevalence for the condition because we don't have very good information for them from the EHR. We applied this approach to an EHR-derived data set from 2 children's hospitals that were participating in a consortium called the PEDSnet consortium. In particular, we wanted to look at children who were at high risk for type 2 diabetes, which if you're not familiar with diabetes in kids, type 2 diabetes is very rare. In children, type 1 diabetes is much more common, type 2 is quite rare, kind of the reverse of what we see in adults. So we identified a set of children aged 10 to 18 years with at least 2 clinical encounters, so some minimum quantity of data available in the database. And on at least one occasion, they had to have a BMI Z-score in excess of the 95th percentile. So that's what we considered as putting them into a high-risk group for type 2 diabetes. And this gave us data from about 55,000 kids across the 2 sites. We look at the distribution of some features that we might potentially use to try to deduce type 2 diabetes status for these children. There are a number of interesting differences between site A and site B. So here I have some features that we might use, biomarkers up here at the top for the subset of children who have biomarkers available, and then a bunch of binary features, including whether or not the child made a visit to an endocrinologist, some prescription medications, diagnosis codes, and then our binary indicators of whether or not the biomarkers were measured. So you can see that there's a difference in the mean glucose, a difference in the mean hemoglobin A1C, suggesting higher distributions, higher values at site B. There's also much more frequent visits to an endocrinologist at site B, greater use of metformin and insulin, much more frequent use of the type 1 and type 2 diabetes codes, and also very different patterns in terms of the frequency with which children are having their glucose and hemoglobin A1C measured between the two sites. So when we look at this overall, what this looks like to me is site B probably has higher prevalence of the condition than site A, but there are also substantial differences in practice patterns and probably also coding patterns taking place between these two sites. So this reinforces concerns about an issue that's often raised in the EHR literature called the transportability or portability problem, that phenotypes developed at one site don't necessarily perform well at another site because each healthcare system has its own unique practices as well as its own unique patient population. So when we did the phenotyping for these two sites, we did it separately within each site in order to acknowledge the presence of these differences. And finally, down at the bottom of this slide, I have the prevalence of type 2 diabetes as captured using the eMERGE Consortium rule-based phenotype. And you can see it was estimated at just 0.3% prevalence and 0.9% prevalence between the two sites based on that algorithm. That's really, really low. That's a prevalence that's more in the range, if not a little bit below what we would expect in a general population. So it's definitely suggestive of the fact that that particular algorithm, which was developed for adults and not for children, is probably not adequately sensitive in this population. So we applied our Bayesian latent phenotyping approach to this data, and this is a set of a sample of posterior estimates from that model for some of the parameters, but not all of the parameters that went into the phenotyping, just to give you kind of a flavor of what you get out. So for things like continuous biomarkers, we get estimates of the mean shift in the biomarker between patients with and without the underlying phenotype. For things that are binary features like codes or prescription medications, we get shifts in the probability, which allow us to estimate the sensitivity and specificity of having or not having those codes. So we can see in the features that I've highlighted here in bold red, there are some big differences in the values that we come up with between the two sites. With things like, for instance, making a visit to an endocrinologist, having a much different specificity between the two sites. Finally, we can estimate the odds ratio associated with having the odds ratio for having your glucose measured associated with having the underlying type 2 diabetes status. And we see that patients with the underlying type 2 diabetes status are much more likely to have their glucose measured than patients who don't have the condition of interest. And that's not at all surprising, but reinforces the sense that we really need to be worrying about this, these potential missing not at random missingness mechanisms in the methods that we're using. And then finally, the most important piece of information that we get out of this phenotyping approach is the posterior probability of having the condition of interest. So here I'm showing the distribution from site A and site B of the individual child-specific probabilities of having the phenotype. What we'd really like to see is this, remember, this is a mixture distribution. We're mixing across the two classes. What we'd really like to see is that these are separated as much as possible. We have one peak close to 0 that represents the children we're pretty sure don't have the condition. And then we have a second peak that's going to represent the kids that we suspect do have the condition. You can see we're getting fairly good separation for site A, a little bit poorer for site B. Site B is also shifted to the right, representing a higher estimated prevalence in that population. But in practice, what we would do is take these probabilities and use them as our phenotypes, potentially as our outcomes in a subsequent study of, say, risk factors for childhood type 2 diabetes. So my general thinking about EHR phenotyping, just like my general thinking about identifying questions that are fit for use, is it all comes back to the quality and the consistency of the data that we have available. So it's just of fundamental importance that we have adequate systematic data capture for the key data elements that are going to become part of our subsequent analysis. The accuracy of the phenotype that we develop is going to be dependent on data provenance, on the constituent data elements that result from that data provenance process. And particularly in EHR data, there's a high risk of differential misclassification if we just use a simple rule-based approach, because of the fact that we have different information available across patients, and those differences are going to be related to characteristics of the patient that may also be related to the exposures or outcomes that we're interested in. And finally, as we saw in the PEDSnet example, transportability of phenotypes may be limited due to heterogeneity of practices. So it's always best practice to validate the phenotype that you're going to use in your specific healthcare system, or to develop the phenotype in your specific healthcare system, and not just to take something off the shelf and assume that it's going to perform equally well in your setting as it did in the setting where it was developed. All right, so the last thing that I want to talk about is, once we've identified a question that EHR are fit to answer, and once we've figured out how we're going to come up with the measures that we analyze to answer that question, we know that there's going to be residual measurement error in the data elements that we've extracted. So what can we do to account for that potential source of information bias? A point that I like to make to all audiences that are considering using EHR data to answer research questions, and a point that I think is particularly important in light of research practices that came up during the pandemic, is that using healthcare data to answer medical research questions creates the possibility of potentially generating very, very precise but biased estimates. So early in the pandemic, when people were getting excited about using EHR data to rapidly generate evidence, I saw this pernicious concept arise that if we have a sufficiently large database, we don't need to worry about this statistics stuff. We don't need to worry about bias. We don't need to worry about methods, because a big enough database will get you to the right answer. And that is terribly false. It just means that we're going to become increasingly certain about our fundamentally incorrect answer. So in this last section of my talk, I'm just going to focus on one particular issue that we might be interested in addressing, with the focus on trying to reduce the bias in our estimates and get closer to that target that we're really shooting for. But in general, I think using methods that specifically address sources of bias in EHR data is absolutely imperative, because they are big databases and they do give us potentially a very false sense that we're coming up with correct answers. So given that we know that the data are error prone, we know that the phenotypes that we've extracted from them are imperfect, what can we do to reduce bias in subsequent analyses using those phenotypes? This is essentially an outcome misclassification problem if the phenotype is an outcome, but there are some specific challenges of the EHR setting, specifically that validation data are expensive and difficult to obtain, typically consist of conducting manual chart review of EHR data. Operating characteristics of our phenotypes may vary substantially between healthcare systems because of the transportability problem. And because of differences in the amount of data and the kind of data available across patients, there's a high risk of differential misclassification. So many methods assume non-differential misclassification. We want to make sure that we are relaxing those kinds of assumptions. I'm assuming that we're in a setting where we would like to make inference about the relationship between some outcome Y and predictor X. And this is taken, the specific applied area this is taken from is that of breast cancer. So I'm going to use as an example a setting where we'd like to look at the relationship between second breast cancer events, or SBCEs, in women with a personal history of breast cancer, and a variety of patient characteristics like age, the stage of their first breast cancer, and their hormone receptor status. Because we're using EHR data, we don't observe Y for anybody. Instead, we just try to make our best guess at it using the data we have available. So instead, we come up with an imperfect phenotype S, and we end up looking at the relationship between S and X instead of Y and X, which doesn't get us quite the parameter that we're interested in estimating. I'm assuming we also additionally have some kind of auxiliary information about the relationship between Y and the phenotype. Here I'm giving the sensitivity and specificity of S indexed by X, so allowing for the possibility that sensitivity and specificity of our phenotype varies by patient characteristics. So there are a bunch of different ways that we could think about using the available data to try to answer a question about the relationship between Y and X. If we're very, very conservative, we don't trust the quality of our EHR phenotype, we could say, well, I'm going to conduct a validation study. I'm going to look at the records of a small subsample of patients. And just in that validation subset, I'll be able to get the true outcome, and I can look at the relationship between Y and X. So the subscript V here is denoting the fact that this analysis is conducted in the validation sample only. It's going to give us a consistent estimate for beta because we have the true outcome Y, but it's going to be really inefficient because we just have a small validation sample. If we're very excited about real world data, we trust our surrogate, we think it's really high quality and we're not too worried about this misclassification stuff, we could look at the relationship between S and X in the full sample. So the subscript F here is denoting full sample. But the problem with that, of course, is that our phenotype is not actually perfect. So this is the high precision, but also potentially high bias setting. We could leverage existing methods for outcome misclassification. So there's a very nice maximum likelihood-based outcome misclassification approach that was proposed a long time ago, which we could leverage to try to reduce bias in an estimate that incorporates information on the surrogate. The limitation of this approach being that we do need to specify a misclassification model. And the results I'm going to show in a moment, I'm assuming a misclassification model that is non-differential. So sensitivity and specificity don't vary according to X. You could actually use a more complex misclassification model, but you do need to specify it. So you need to be able to correctly specify the misclassification model. I'm going to contrast this with an approach which was developed by a student at Penn, Jesse Tang, who was working with me and a colleague, Yang Chen. The method that Jesse developed was an attempt to adaptively weight our estimate based on the validation data only and our estimate based on the full sample using the surrogate, where the weighting is going to reflect the quality or the accuracy of our surrogate. The nice thing about this approach is that it doesn't assume non-differential misclassification, so it can handle differential misclassification. And it also doesn't require explicit specification of a misclassification model. So the weighting is going to be data adaptive. In order to do this, we define an additional regression model, which is the regression of the surrogate outcome on X in the validation sample only. And that's going to allow us to construct this estimator, which I'm going to call the augmented estimator, which is basically a weighted, so this is my heuristic way of thinking about this at least, it's basically a weighted combination of our estimate based on the validation sample and our estimate based on the surrogate in the full sample, where the weight that we place on each of those two things is guided by the strength of the correlation between our estimate in the validation sample using the surrogate and our estimate in the validation sample using the true outcome Y. So essentially, if those two estimates are really highly correlated, this correlation matrix in the middle of this equation will be very close to 1. Beta hat V and beta hat star V cancel out, and we put all of our weight on beta hat star F, our estimate in the full sample, because that would be strong evidence that our surrogate is pretty good. On the other hand, if we take a look at our estimate based on the surrogate and our estimate based on the true outcome, and they're largely uncorrelated with each other, then this would be essentially a matrix of zeros. The second term cancels out, and we end up just placing all of our weight on beta hat V, our estimate using the true outcome. So we compared the performance of these 4 different approaches in a data set derived in this setting of looking at second breast cancer events in women with a personal history of breast cancer. And in this setting, we knew the truth. We had the true outcome based on validation data for everybody. So the dotted black line here represents the estimate that we got using the truth in the full sample, along with its confidence interval in the gray shaded bands. We then took a look at the performance of the 4 approaches that I described as we varied the size of the validation sample. So the ends here are the size of our validation subset. And as we varied the quality of our EHR-based surrogate. So the upper left, which is called the high specificity surrogate, is a really accurate surrogate. The upper right is a lower accuracy surrogate that demonstrates non-differential misclassification. And the lower left is a lower accuracy surrogate that exhibits differential misclassification. So the basic quick summary take home of this figure is obviously using only the validation data gives you an imprecise estimate that's kind of unstable when you have a small validation sample size, but that obviously gets better and is going to converge to the truth as your validation sample gets bigger. Using only the information from the surrogate, but in the full sample, gives us an estimate that's very precise. So model 2 is always going to be very precise, but how close it is to what we would have gotten using the truth depends on how good our surrogate is. When our surrogate is very good, it's quite close. When our surrogate's rather poor, it's farther away. Model 3, which makes a maximum likelihood-based misclassification correction, performs well when we correctly specify that misclassification model. So as I said, we're assuming here non-differential misclassification. So when non-differential misclassification holds, everything looks good. When it does not hold, using that misclassification correction approach actually exacerbates the bias relative to the uncorrected approach. And finally, the augmented estimator is going to give us something that is intermediate in terms of its precision between the validation data only and surrogate estimate, because it's a weighted combination of those two things. It's going to get us closer to the estimate based on the truth in most settings, but it can be a little bit unstable when we have a small validation sample and a complex misclassification process. So I don't believe in anything that's a free lunch. This is not a magic bullet that works in all settings, but it does work across a lot of settings and does allow us to relax the assumption of non-differential misclassification. All right, so I'm going to wrap up so that we have a couple minutes for questions. I actually think I'll skip this slide and go straight to my conclusions. Especially in medical research, but I assume in other areas of application as well, there's often this feeling of urgency. We need the answer right now. We can't wait for the clinical trials. You know, we have to do the best that we can with the data that we have available. And real world data certainly provide an opportunity to address those kinds of urgent questions. But sometimes it's just really not a good idea because the questions that we would like to answer are just not appropriate to answer using EHR data. And the odds of getting to a correct answer are actually quite slim. And when we come up with erroneous answers, biased answers, we're not advancing clinical care. We're not advancing the medical research base by promulgating low quality research. Understanding the data sources that we're using, whether they're medical or other settings, is really important. Understanding where the data came from, how it was generated, and what the implications of that process are for things like missing data mechanisms and measurement error are really key. And the final point that I always like to make, and especially to statistical audiences, is that it's so important for us to have a seat at the table in this type of research, because other fields may be less aware of the study design and bias issues associated with using these data sources, and are willing to just rush forward with technical advancements that allow them to use the data, but not necessarily to generate valid answers. So participating, not tuning out because we think the data is poor quality, is really, really important. And helping the scientists and medical researchers and public to feel that we are engaged, that we are trying to help them reach the best answer that we possibly can, and not just playing traffic cop and saying no and pointing out all the things that are bad about the data, I think is really, really important. I'll end by just acknowledging my collaborators at Penn and the financial support of the Patient-Centered Outcomes Research Institute. And you can always reach me by email if we don't have time to answer your question this afternoon.